Okay. Welcome to the HNRCA seminar series this week. I'm Roger Fielding. I'll serve as your moderator. I just want to remind everybody that's joining the seminar to use the Q&A function below to ask questions, and we'll have time at the end of the seminar for questions and answers. So without further ado, it's my pleasure to introduce Dr. Bryan Bergman, who's a professor in the Division of Endocrinology, Metabolism, and Diabetes at the University of Colorado Anschutz Medical Campus. Dr. Bergman uses deep phenotyping and tissue sampling from humans across the spectrum of metabolic health to understand mechanisms explaining muscle insulin resistance, development of steatotic liver disease, and tissue-tissue crosstalk. Much of his work focuses on lipids stored within skeletal muscle that decrease insulin sensitivity, as well as adipose tissue, paracrine, and endocrine signaling to impact function of local and distant tissues. Dr. Bergman has been continuously funded by the NIH for the past 18 years and has two active current R01s, along with collaborations with industry. So his presentation today is going to be intramuscular adipose tissue paracrine signaling impacts on skeletal muscle. Welcome, Dr. Bergman.
Hey, thank you, and thank you for the nice introduction, and thanks for having me. Appreciate you having an opportunity to come virtually and chat with everybody. All right, so I always like to give just a brief big picture about what my work is all about. And the overall goal, kind of the end all be all for my work, is to develop therapeutic targets to prevent or treat type 2 diabetes. I focused on muscle because muscle disposes of 80% of a glucose load after you eat a meal or drink a glucose-laden drink. So it's by far the biggest sink for glucose disposal, and yet we really don't have good therapeutic targets directed specifically at muscle. We know that metformin has some off effects, but mostly it's weight loss and exercise training that does the heavy lifting with muscle insulin sensitivity. Weight loss is getting a little bit easier with the new obesity medications, but exercise is still really hard for people to stick with. And so our goal is to try to figure out how to make muscle more sensitive to insulin to prevent and delay prediabetes and type 2 diabetes. So we do that a number of different ways. As was just mentioned, we look at a lot of lipids stored within muscle cells, and we've pushed the field forward by figuring out where specific species of lipids are stored and how that's related to diabetes risk, and then how those lipid species change after insulin sensitizing interventions. And then what I'm talking to you about today is this whole concept of intramuscular adipose tissue, so the marbling of fat around muscle and how that's related to diabetes risk, and we'll get into that here in a moment. And then we're also starting to look at some other interesting questions and kind of push the lab in new directions. One is, how does visceral adipose tissue impact metabolic health? I think most people think that visceral adipose tissue is really well phenotyped and well known, and we know everything about it. But when you dive in the literature, the exact opposite is true. I think most people just think of inflammatory cytokines. A lot of people just measure TNF-alpha and IL-6 and call it good. I think there's a lot left to know and learn about visceral adipose tissue and how it impacts metabolic health. We're looking at how visceral adipose tissue is impacting fatty liver disease with mast cell to MASH, because visceral adipose tissue drains into the portal vein, directly hits the liver, and can have really potent impacts on liver because of that direct connection. So we're starting to study that. And they're also looking at how adipose tissue and muscle are crosstalking with one another and how that is changing each one of their endocrine and paracrine signals and how that's related to metabolic health. So that's just a brief overview of the lab and what we're doing. But let's dive into the intramuscular adipose tissue story here. So first, let's just talk about it in a little bit more detail. So this is a CT of somebody's thigh. This is an overweight or obese individual. And you can see muscle is shown here in green, subcutaneous adipose tissue in red, and then this yellow marbling is the intramuscular adipose tissue. And so all of our muscles have IMAT. Um, and it is in every one of the muscles that we've been able to study so far. And no matter if you're lean or obese or overweight or in between, everybody has a little bit of this. Importantly, it's not lipid stored within muscle fibers itself. People get that confused because we've spent a lot of time studying that and talking about it. This is actual adipose tissue and all the cells that are associated with adipose tissue that is underneath the muscle fascia and laced between muscle fibers and muscle groups. And we all know this depot. This has been the, I think, a depot hidden in plain sight our entire lives. So when you look at a steak, all that marbling makes your steak taste great. Yet it's probably not so good for the cow's health, and it's not good for your health when you have a lot of it in your muscle. So it's the marbling of adipose tissue in muscle. This is what it looks like when you take a muscle biopsy. We've actually developed an ultrasound guided biopsy to specifically target IMAT. And this is what the biopsy looks like when you put it on a dissecting scope at 30 times X. And you can see the muscle up here in red. On the left-hand side, there's some fascia. And on the right-hand side is the IMAT itself, or the adipose tissue. And you can see the little balls, those little adipocyte cells. After you dissect them apart, which we do for all of these to make sure that we're looking at just IMAT and not kind of a mix of all the tissues, you can now see IMAT isolated on the left and muscle on the right. So this is how it looks like when you take a biopsy from a human participant. So why should you care? So here is the first data that really put IMAT on the map, and this is from Brett Goodspaster in 2000. So on the y-axis, we see insulin sensitivity from an insulin clamp. So high numbers mean high insulin sensitivity, and then the opposite, of course, for low numbers means low insulin sensitivity. On the x-axis, we have subcutaneous adipose tissue, also measured by CT. And you see what you know already, and that is that it's subcutaneous adipose tissue is a healthy adipose tissue depot. It's a good place to store excess calories, and it's unrelated to whole body insulin sensitivity. However, when you look at IMAT, you get a very different story. And if you look at the x-axis, the more IMAT individuals have, the less insulin sensitive they tend to be, and vice versa. So this is the first data that really showed that there was a strong relationship between IMAT and insulin sensitivity that really got people fired up and excited about studying it. Since then, we've learned quite a lot. One, we know that IMAT accumulates with obesity, and it can accumulate very quickly with immobilization studies. If you put somebody in a foot down cast or a hind limb immobilization experiment, IMAT accumulates quickly, and muscle mass decreases a little bit, but the strength that goes down associated with immobilization seems more related to the accumulation of IMAT. This has been studied, IMAT has been studied in many different populations now, and it always seems to explain insulin resistance associated with age, sex, and ethnicity. No matter what group is studied, and it really always seems to be related. So exercise, it's my sort of view of the world that exercise is this pluripotent, amazing thing for all tissues, but it really doesn't seem to do a lot for decreasing IMAT content. It more seems able to decrease accumulation of IMAT over time. Although there is some data on resistance training coming out to show that resistance training actually might be more potent than whole body aerobic activity to decrease IMAT content. The one interesting part is that IMAT is a smaller depot than visceral adipose tissue when you add it up, and yet the cardiovascular disease risk for each one of them is quite similar, and it just shows the potential potency of IMAT and how it might be related to metabolic risk. And we all know that as well that IMAT is related to sarcopenia. As people get older, they tend to have reduced muscle mass, and that muscle volume is almost replaced by intramuscular adipose tissue. And so it's heavily involved with aging and aging-related sarcopenia and also obesity sarcopenia. But the problem with the field up to a few years ago is we knew very little about how IMAT could be influencing metabolism. Were these things true, and unrelated? Or was IMAT actually causing some of these problems? And that's really where my lab tried to pick up this this research area and try to push it forward. So the way we approach this is sort of a very simplistic way of looking at the world. And that is, if IMAT is right next to muscle, maybe what's going on is that it is having a lot of paracrine secretions and secretome in general that's coming out of the IMAT that hits the muscle and causes muscle problems. So that is what we decided to chase down. So when we have individuals in our studies, we collect IMAT and we look at it on a dissecting scope, like I showed you before, and we tease apart the muscle from the intramuscular adipose tissue. And we're also in the operating rooms with people getting elective laparoscopic surgeries, whether it's a hernia operation or a bariatric surgery, anti-reflux, etc. And from those individuals, we're also collecting subcutaneous and visceral adipose tissue against which we can compare because we know subcutaneous is a healthy depot, visceral is a bad depot. And the question we had that NIH really wanted us to address is how bad is IMAT relative to these other well-studied adipose tissue depots? So the way we've approached this is we take these biopsies back to the lab. We keep them in DMEM that's warm at 37 degrees C to not temperature shock the tissue. We cut it up into small pieces, rinse it, filter it, weigh it, and then we put it in culture in a ratio of 20 microliters per 1 milligram of adipose tissue. And then we culture it for 24 hours after a 1 hour rinse and collect all of what is secreted so we can actually try to quantify and look at all of these paracrine factors that are coming out of IMAT and related to these other depots. So importantly, when we're thinking about adipose tissue secretome, we really need to think briefly about the composition of adipose tissue. And most of us know this, but adipose tissue is not just adipocytes. It's a lot of other cells that are existing in adipose tissue, like immune cells, whether they are Tregs or macrophages. There's a lot of extracellular matrix and fibroblast, progenitor cells, etc. And we know based on single cell RNA-seq studies that when individuals progress from a healthy to a less healthy or more metabolic disease state, the composition of their adipose tissue changes dramatically. And there's a lot more immune cells that are more M1 polarized. And I know that terminology is not really loved by everybody, but there are more inflammatory macrophages, a lot more extracellular matrix, etc. And this is really important because it always blows my mind to say this. 90% of all the proteins that are secreted by adipose tissue actually come from the non-adipocyte portion of adipose tissue, i.e., the stromal fraction. All these other cells we just talked about do most of the heavy lifting when it comes to what is secreted from adipose tissue. That means that the composition of adipose tissue really has a huge impact on the secretome and really impacts the paracrine signaling that's coming out of these depots. So when we look at our secretome, we have decided to try to keep all of these cells together, keep the adipose tissues together as a tissue and studied secretome rather than a more reductionist approach where people have collagenase dissected adipose tissue to just pull out adipocytes and look at what the adipocytes are secreting because we want to know what this tissue is behaving at or how it's behaving in the in vivo setting. And this is actually really important when you try to make some conclusions about what's going on in vivo. This is data from John Fain at Tennessee from a while ago now. But what they did is they did this exact comparison I'm talking about. They looked at what was secreted from the tissue on the left-hand side compared to adipocytes isolated from the same tissue. And it doesn't matter which signaling molecule you want to look at, whether it's prostaglandins or IL-8 or adiponectin, TNF-alpha, etc., what you can see, and we can just look at IL-8 here, what the tissue secretes is dramatically different than what the adipocytes secrete. And this just reinforces the point that not only are the other cells, i.e., the stromal cells in adipose tissue, doing a lot of the heavy lifting when it comes to the secretome, there's also cell-cell communication within the tissue that actually allows the secretome to occur. So we've decided to study the tissue as a whole to try to better mimic in vivo physiology. So one of the first questions we had was to try to answer this question, does IMAT actually cause insulin resistance? And so what we did is on the y-axis here, we have insulin sensitivity in vitro. This is a insulin-stimulated glycogen storage assay. And we have a control condition where these cells, so these are primary myotubes in vitro. We just give them DMEM, so a normal cell culture medium. And it's at 10% of the total conditioned media, which is going to match the conditioned media or the secretome that we give from these other depots. And because these primary cells are variable in terms of their insulin sensitivity, we normalize every experiment to 1 based on this control condition. When you look at the subcutaneous adipose tissue, you can see that there was really no change in insulin sensitivity when we gave 10% of its secretome as conditioned media to these cells. And that again just reinforces what we know. subcutaneous adipose tissue is a healthy adipose tissue depot and a good place to store excess calories. Then we gave from these same donors visceral adipose tissue conditioned media to these cells, and what you can see is a dramatic decrease in insulin sensitivity. This is again not surprising. This is expected. We know this because we know that visceral adipose tissue is a negative adipose tissue depot. What we didn't know was how potent was IMAT. And this was a really important question to answer to figure out if IMAT is just true and unrelated to diabetes risk or if it's actually a causative agent. And what we found was that it was very similar in terms of its potency to visceral adipose tissue. So this actually says that everything that's being secreted out of intermuscular adipose tissue can actually cause decreased insulin sensitivity, at least in vitro. The downsides with these data were that the subcutaneous and the visceral adipose tissue came from different donors. Uh, they came from bariatric surgery participants, whereas the IMAT biopsies came from participants in our other studies that we had going on. So it wasn't the best comparison. And in fact, it probably was the least powerful comparison for IMAT because the individuals from whom we generated these IMAT biopsies were actually less obese and more metabolically healthy compared to the others. So we really wanted to study this further in paired individuals. So that's what we're doing. And we just finished this study. And it's a really fun study because my team gets to go into the operating rooms for people that have been consented to have these elective laparoscopic surgeries where we actually collect these biopsies. And you know you're a tourist in the OR when you're taking pictures of each other. We're definitely those people, but we're having a lot of fun doing it. So in this study, we consented 102 individuals. They spanned the BMI range of 20 to 50 and also spanning the range of insulin sensitivity. And we have a collection of tissues in the OR. And it's actually really wild. So the way this worked was as soon as the patient would go down for anesthesia, our team would rush in like a Formula One pit crew, and we would do this ultrasound guided biopsy in about 2 or 3 minutes because you absolutely can't delay surgical time because that gets really expensive. So we took a vastus lateralis IMAT biopsy, similar to many other of the biopsies that are taken for research, so we can collect the IMAT. While we were in there, we were able to collect some other depots that allowed us to answer some really interesting questions. We also were able to sample some muscle from the transverse abdominis. So this is right from the laparoscopic incisions. So we pulled out some of that muscle and also took it back to the lab and dissected it. And this was allowing us to answer some really interesting questions like, does upper versus lower body obesity and lower body adipose tissue differentially impact metabolic risk? We know from a lot of work that's out there, like Michael Jensen's data in particular, that upper body obesity seems to be more negative compared to lower body obesity, and the cells from the upper body adipose tissue seem to be actually worse in terms of their impact on metabolic risk. So this allowed us to answer that question with IMAT as well. We also were able to obtain superficial subcutaneous adipose tissue, again from the laparoscopic incisions. We were able to also collect visceral adipose tissue as well from these individuals against which to compare for a known negative adipose tissue depot. And then we were also able to collect liver biopsies to kind of answer this question or start to answer this question about portal drainage of mesenteric and visceral adipose tissue and how it hits the liver. So a lot of fun side projects have come out of this work. All right, so let's dive into this. All this data is unpublished, so we're hoping to get it out here pretty soon. The first question we had or we wanted to answer was, is all IMAT created equal? And so what we did is we took those paired samples I was mentioning, of vastus lateralis and transverse abdominis. We actually looked at their ability to decrease insulin sensitivity in vitro. So on the y-axis here, we have this insulin sensitivity assay in vitro. Again, it's insulin stimulated radioactive glucose storage in the glycogen. So it's a very functional assay. And again, the control condition here is just DMEM on these cells. And these are L6 myotubes. And we wanted to minimize donor variability in terms of its response to conditioned media and just focus on the conditioned media. So we've chosen L6s. And what you can see is whether you're looking at vastus lateralis IMAT here in the light gray or transverse abdominis IMAT. And the darker gray, they're equally potent in terms of their ability to induce insulin resistance. And this is actually sort of fascinating, and it implies that it doesn't matter whether your IMAT's in the upper body or lower body or where it's probably distributed into the body. It seems to be similarly negative in terms of diabetes risk. And that maybe shouldn't surprise us when you think about the microenvironment in which IMAT exists. It's confined by muscle. We know that confining adipose tissue tends to be more negative when it comes to its metabolic risk. And so the microenvironment of the IMAT in the muscle is probably very similar no matter where it is in the body. And this was our data suggesting that's true. So based on that, just to make our comparisons a little easier, we combined these groups and just had IMAT as one group instead of two. And so now we're looking at paired IMAT, subcutaneous, and visceral adipose tissue samples from these individuals. I think this is 34 or 35 people that we did this on. And so again, this is this nice 30% or so decrease in insulin sensitivity as a result of just a very small amount. This is 10% of IMAT conditioned media exposed to the myotubes for 3.5 hours during a serum starve. So I think it's a realistic dose of the paracrine signals that that muscle would see in vivo. We then compared that to subcutaneous and visceral adipose tissue from the same individuals these times, and the data are, I think, just fascinating. What you can see is subcutaneous adipose tissue actually has a positive impact on insulin sensitivity. And the spread here is pretty dramatic, and we can kick around that if you want. But what it shows is that the secretome of subcutaneous adipose tissue is actually beneficial on a population average. And it's definitely much more beneficial compared to IMAT. When you look at visceral adipose tissue, we have a negative impact of visceral adipose tissue conditioned media on this myotube insulin sensitivity, and it is less dramatic, again, in these paired individuals this time, compared to intermuscular adipose tissue from the same individuals. So this says that IMAT actually is more potent than visceral adipose tissue in terms of its ability to decrease metabolic function, in this case, insulin sensitivity in vitro. We then looked at our donors based on whether they were normal glucose tolerance or if they had prediabetes or type 2 diabetes. Again, y-axis is the same. Now we're looking at this merged IMAT. And what you could see is no matter who you are, if you have IMAT, it is bad for your muscle insulin sensitivity. So in normal individuals with normal glucose tolerance, kind of across the lean, overweight, obese continuum, IMAT decreased insulin sensitivity, and it was a little bit worse. It was significantly worse in individuals that already had prediabetes or type 2 diabetes. And so IMAT seems to always be bad in terms of its risk for insulin sensitivity. We did the same thing for visceral adipose tissue from the same individuals, and I found this pretty fascinating, that if you're an individual with normal glucose tolerance, your visceral adipose tissue didn't change insulin sensitivity when it's administered in that way. It was only if these individuals had prediabetes or type 2 diabetes that you saw a significant decrease in insulin sensitivity. So this to me implies that IMAT is always bad, whereas visceral adipose tissue might not always be bad if you're a more healthy individual. And it's I think really powerful because that says that conditioned media is always being exposed to your skeletal muscle, causing decreased insulin sensitivity. We also looked at our population just based on age, everybody combined together. Again, insulin story or insulin sensitivity, the y-axis, age on the x-axis, we have this inverse relationship. It's significant. We compared that to visceral adipose tissue. There's still a similar relationship with visceral adipose tissue. It wasn't significant in these 33 or 34 individuals, and the slope wasn't quite as strong. Again, just suggesting that as people age, it looks like as a population level that their IMAT tends to be more negative when it comes to decreasing insulin sensitivity in muscle. So we know the IMAT secretome is more potent than VAT, and it's exacerbated by age and type 2 diabetes. But the biggest question really is why? What's going on? What's the change here? And the lowest hanging fruit, when you think about how adipose tissue impacts metabolism, is inflammation. So we looked at the inflammatory response in these tissues or by tissue. And this is again L6 myotubes to eliminate donor variability between the response to the conditioned media. And we administered the conditioned media. This was 10% for conditioned media in terms of the percent of all the media that these cells are being bathed in for 15 minutes. So it's a really acute response. And we're looking at the inflammatory response. In this case, we're looking at phospho to total ratios. And this is a junk activation. And you can see compared to this dotted line, which represents the normal control condition with DMM only, both subcutaneous and visceral adipose tissue increased inflammation. But in this case, the transverse abdominis IMAT increased more dramatically than those other two, implying that IMAT is actually more inflammatory than both subcutaneous and visceral adipose tissue. It then didn't matter which inflammatory pathway we looked at, whether you're looking at phospho-P38 or if you're looking at NF-kappa B. In this case, the IK beta alpha, when it goes down, it means there's more activation. It's one of these weird markers. But it shows that no matter what the inflammatory pathway, IMAT was more inflammatory compared to subcutaneous and visceral adipose tissue. Same here with phospho-ERK. Again, more inflammatory from IMAT compared to the other depots. So this is likely one of the mechanisms by which IMAT is decreasing metabolic function or muscle insulin sensitivity. So we know IMAT's more inflammatory than SAD or VAT. The data I'm not showing you, but is also true, is that the IMAT inflammatory signaling, it really didn't seem to change with age, prediabetes, type 2 diabetes status, or BMI. So it really can't explain why IMAT seemed to be more negative when individuals were more overweight or had type 2 diabetes or were older. And so there's clearly something else going on. And so we were looking at other secreted proteins and what those proteins might be, and if they can give us insight into how IMAT might be working. So the way we've done this is we take IMAT and we take that conditioned media and we sent it off for O-link analysis, along with paired subcutaneous visceral tissues as well. And we're trying to answer the question why IMAT is more metabolically potent than visceral adipose tissue. This is just some prelim data because we're still going through this data. We have some batch effects that we're working out right now, but it just shows a volcano plot of differences between the vastus lateralis, IMAT, and visceral adipose tissue protein secretion. You can see that there's a number of proteins that are upregulated and a number that are downregulated. And we're going to try to dive in here to try to figure out what could some of these proteins be that are making IMAT worse than visceral adipose tissue when it comes to metabolic risk. And I think there's going to be a lot to learn there. We've already done some of this work with a collaboration with Eli Lilly, and this is what we call internally as our novel protein discovery project. We took IMAT biopsies from individuals and we made conditioned media, just like I've been mentioning to you. We then did proteomics on that conditioned media. And importantly, we then correlated the protein abundance to donor insulin sensitivity measured by an insulin clamp. So now we have this list of proteins that's related to donor insulin sensitivity. What we did then, we then filtered those proteins for having a signal peptide sequence so that we know that they were actually secreted and weren't just there because the cells were damaged. We purchased those proteins in a recombinant way, or just recombinant proteins. We administered them to primary muscle cell cultures with a dose response, and then measured the change in insulin sensitivity. And the goal here is to go from observation to actual causality for individual proteins in culture of how IMAT might be working to decrease insulin sensitivity. So we tested 12 candidate proteins because they're really expensive and you can't do more than that. That was already a lot. I had to shoot the lock off my wallet to get this study done. We found that 6 caused insulin resistance in vitro. And so I was thrilled to be batting 500 when it came to finding proteins that actually caused decreased insulin sensitivity. I'm going to tell you a story about 2 of them, and that is plasma and cellular fibronectin. So this is data from L6 myotubes, which is how we did the screening. And this is, again, our insulin sensitivity measurement in vitro. And you can see both plasma and cellular fibronectin decreased insulin sensitivity. So it's interesting, the proteomics output just said fibronectin, and it didn't discriminate whether it was plasma or cellular. Plasma fibronectin is thought to come mostly from the liver. Cellular fibronectin is most likely what any adipose tissue depot, but in this case, IMAT is going to be secreting locally on myotubes. We then repeated these data with primary myotubes to make sure that it was legit and real. And sure enough, both plasma and cellular fibronectin decreased insulin sensitivity in vitro. We then went and looked at some of these circulating concentrations to make sure that they kind of also just jived in terms of our other populations that we have and how the circulating concentrations related to insulin sensitivity. And so this is a group or a cohort of people that we study quite a lot. They range from athletes all the way up to individuals with type 2 diabetes. So they effectively span the physiological range of insulin sensitivity. And what you can see is the plasma concentration of plasma fibronectin here on the y-axis. And you can see there's this inverse relationship to insulin sensitivity. Same is also true when you look at cellular fibronectin. Cellular fibronectin tends to be lowest in most insulin sensitive individuals, in this case, athletes, and it tends to be highest in people with type 2 diabetes. So we thought this was at least confirmatory that this could be playing a role in terms of its relationship to insulin sensitivity. But then to prove this, we really wanted to figure out, okay, let's make sure that we know that fibronectin is acting on myotubes specifically. The literature says that fibronectin acts through this integrin receptor, alpha 5 beta 1. So we CRISPR knocked down the beta 1 component of the integrin receptor, and we also chose a pharmacologic antagonist of alpha 5 beta 1 as well to test this out. And so what we did here is we have our insulin sensitivity axis or, assay again here on the x-axis. We have our scramble control from the CRISPR knockdown as our 100%. When we took our scramble control cells and gave them cellular fibronectin, they were still had a decreased sensitivity to insulin as you would expect. So just the CRISPR itself didn't have a negative impact on the response of these cells. When we knocked out the integrin beta 1, it actually seemed, if anything, kind of increase a little bit, but there was no significant change in the insulin sensitivity just by knocking out the integrin receptor. But when we gave the cellular fibronectin to these integrin receptor knockout cells, there was no change in insulin sensitivity, and we restored insulin sensitivity compared to the scramble control, meaning cellular fibronectin is certainly acting through integrin alpha 5 beta 1 to decrease muscle insulin sensitivity. We then repeated this with our pharmacologic antagonist as well. This was K34C. It's a well-known antagonist, and we found the exact same results. A nice decrease with vehicle control when cellular fibronectin is administered. K34C on its own doesn't do anything to insulin sensitivity, but it prevented completely the decrease in insulin sensitivity when you administered cellular fibronectin. So we're now walking down the road towards the drug discovery here, which is super exciting. And this is in collaboration with Eli Lilly, who clearly know what they're doing. The next step here is now we're going to administer the cellular fibronectin to animals, make sure or see if it decreases insulin sensitivity. Then we're going to try to electroporate siRNA against this integrin alpha 5 beta 1 to try to see if we can restore functionality in vivo. And if we do, we're off to the races in terms of having a potential pharmacologic or therapeutic target that we could use to antagonize the IMAT secretome effect on muscle insulin sensitivity. So we're really excited about these data, and we're now trying to chase down some of our other proteins that we think might be causing some of these problems. Okay, briefly, we need to just chat about cell composition of IMAT because one of the biggest questions we have is the composition of IMAT malleable? Because that would mean that the secretome is malleable, which means that lifestyle interventions might be able to change the secretome and improve the secretome in general and improve or decrease IMAT's ability to make muscle less sensitive to insulin. So we did a study. This is a combined pre-post intervention where we had combined weight loss and exercise training in individuals mostly with pre-diabetes. So they were low insulin sensitivity. And we did single nucleotide RNA-seq. So this is pooled data from about 8 or 10 donors before the intervention and pooled data from 8 or 10 donors after the intervention in terms of single cell RNA-seq. And what you can see are a couple interesting things. The pre-adipocyte 2 population decreased quite a lot after the intervention. This is interesting because we know pre-adipocytes actually secrete a lot of extracellular matrix, and it could be the cell responsible for the fibronectin. We also see an increase in immune content, which is kind of surprising, but I'll tell you what that's all about here in a moment, and also an increase in the vascular cell component. when you look at these in terms of just percent of total, it shows the data a little bit more clearly. What's not obvious from just looking at the figure above is the FAP, or the fibroadipogenic precursors, increased after the intervention. This is a cell type from which most people think IMAT comes, and it's a less committed cell type in terms of relative to a preadipocyte, which we know is going to turn into adipocyte. FAPs can turn into preadipocytes into adipocytes themselves, but they're also just a less committed cell type. This pre-adipocyte 2 decreased dramatically after the intervention, which is interesting because, as I mentioned, they can do a lot of secretion of extracellular matrix. Vascular cells increase, which might mean that there's less hypoxia and it's a more healthy adipose tissue depot. And the immune cells increase, which initially I thought, how's that even possible? We know that the IMAT is better for you, at least after these interventions. And so how is it possible that the immune cells are increasing? And what we did is we then looked at the gene expression associated with these macrophages, and it looks like after the intervention, they're much more resident or anti-inflammatory macrophages, and much less of these lipid-associated macrophages that are more kind of inflammatory and one polarized type of macrophages. So the good news here is that the IMAT depot is malleable. So it's not just always bad, and it's how much of the depot that you have that dictates its negative metabolic impact in terms of diabetes risk. It's not only content, but it's also the composition. So the cell types seem like they're changing after these combined weight loss and exercise training interventions, which means we can intervene and make IMAT less negative for metabolic health. So now I wanted to pivot away from insulin sensitivity and talk a little bit about sarcopenia. We've been pushing this story forward to try to look at what's going on with IMAT and sarcopenia and advance the field a little bit. So we know, as I mentioned in the introduction, that IMAT tends to accumulate in older individuals compared to young individuals. This is an MRI of a young, lean individual. That would be my 26, so I guess they're overweight. But you can see there's not a lot of IMAT there. Compare that on the right-hand side to an older individual, same BMI, same thigh circumference. So by all means, it looks like they're kind of the same metabolic health, yet there's much more IMAT marbling. And this just shows the age impact on muscle quality, and that IMAT is actually increasing over time on a population level in terms of most individuals. And this is related to decreased muscle strength. And what's wild is Goodpaster has actually done a bunch of this work, as have others, that when you look at the force that's generated, even normalized per cross-sectional area, if you have more IMAT in that muscle, the muscle is generating less force, even when normalized for cross-sectional area, which is kind of crazy because in exercise physiology, we're taught that force production is directly related to cross-sectional area. And this is saying that there's some other effect going on where IMAT seems to be making muscle less strong. Given the data that I've shown you, it probably won't surprise you that I think that what's going on is the IMAT secretome might be bathing the muscle with factors that are actually decreasing its ability to generate force. And that might explain these relationships in the literature for the relationship between IMAT and force. We're looking at these IMAT content via MRI with an active study. This is a weight loss study. Right now, these data are just the pre-intervention IMAT amounts on the y-axis here. It's taken by ultrasound. We're getting the MRI data soon. I just don't have it quite yet. And then we're looking at the relationship between IMAT content and strength in a number of different parameters. Isokinetic on the left, and then 60 degrees and 80 degrees per second, 180 degrees per second, excuse me, in terms of isokinetic dynamometry, and also just hand grip, which is sort of the classic way to measure muscle strength. And what you can see is the strong inverse relationship. And this is independent of muscle size. This is just the amount of IMAT that is in somebody's thigh in this case seems to be really strongly related to the force that that muscle can generate. And so now we're very excited to see after these individuals lose weight, does the slope of these relationships change? Is the muscle able to generate more strength? And that's where we're going with this. So one kind of low-hanging fruit again when it comes to how IMAT might be related to muscle strength is myostatin. Myostatin is a myokine secreted by muscle. We know it binds to activin 2 receptors and promotes a loss of muscle mass. NASA is chasing this down as a way to try to prevent the loss of muscle mass that occurs during space flight and unweighting, as a way to prevent all the atrophy that occurs. We wanted to look at myostatin secretion from IMAT to think, okay, maybe IMAT is secreting myostatin locally, and that's having some negative impacts on muscle mass and muscle strength. And sure enough, that's what we found. And this is again is unpublished. No, this is not out there in the literature, but it shows that GDF8, also known as myostatin, is secreted almost twice as much from IMAT compared to subcutaneous and visceral adipose tissue. These are other activin 2 receptor agonists, GDF2 and BMP6. So it looks like IMAT actually could be a pretty potent local source of activin 2 receptor agonism that could be bathing in the muscle and having these impacts to promote sarcopenia, loss of muscle mass, loss of muscle strength, etc. What is also interesting, and this is just an N of 5, and these were data that we mostly generated for a grant, that in older individuals, it looks like there was a trend with an N of 5. This is actually, I think, somewhat compelling that there was more myostatin secretion from IMAT in older compared to younger individuals. Again, suggesting cell composition might be changing in these individuals as they age, and that could be related to the decreased muscle mass and decreased muscle force production. So this is data that we just generated last month, and it's super exciting. And what you see is myotubes that are grown on a slide. These are L6 myotubes again to kind of just eliminate donor variability. They're a control condition on the upper left-hand side, myostatin treated cells on the upper right-hand side, visceral adipose tissue treated cells lower left-hand corner, then IMAT lower right-hand corner. And we know from work from Vanessa Pellegrinelli that visceral adipose tissue secretome can decrease myotube diameter and contractile protein expression, but what we don't know is what's going on with IMAT. We were pleased to find that these data show that IMAT's actually really potent when it comes to its ability to decrease muscle mass and strength, again in vitro. But you could see a nice decrease in the average myotube diameter when these cells are exposed to myostatin. It's actually slightly more potent when exposed to visceral adipose tissue. And again, this is 10% conditioned media. This is for 48 hours to have a little bit more of a chronic effect. But IMAT is the most potent in terms of its ability to decrease myotube diameter. And this is, of course, just showing that IMAT, it actually could be causative in terms of having profound local influences on muscle mass, myotube strength, etc. We also then looked at contractile protein expression to see if IMAT might be having any unique effect. And here we're looking at muscle myosin heavy chain 2. And this is protein expression from a Western blot on the y-axis. Control condition is again just DMEM given to these L6 myotubes. And what was sort of surprising is that it didn't really matter where or what the depot was from which the adipose tissue conditioned media came. Adipose tissue conditioned media decreased myosin heavy chain expression, whether it was from subcutaneous, visceral, or intermuscular adipose tissue. And these are from paired individuals. So all the depots came from the same person. And here's our individual data here on the right-hand side. We also looked at troponin T. And troponin T also had a similar decrease no matter what the adipose tissue depot. And again, our individual data on the right side. So initially, when I saw this, I was kind of disappointed because of the data that I showed you before. It looks like IMAT really could be having a unique impact on muscle in terms of contractile protein content. And what this says is that it's not unique compared to the other depots, but what is unique about it is of these depots, IMAT is the only one that is right next to the muscle. And so while subcutaneous and visceral adipose tissue could have these negative impacts on muscle mass or this in vitro surrogate here from contractile protein expression, they're not going to have a very potent impact because their secretome gets dumped into the systemic circulation and it just then hits the muscle and it is very diluted relative to what happens with intramuscular adipose tissue, where its secretome bathes the muscle in a much more higher concentration. So I think what these data show is IMAT is not unique in terms of its ability to decrease sarcopenia or decrease contractile protein expression, but what's unique is its physical location, and that secretome can then be hitting the muscle, having negative impacts on muscle mass. So to wrap things up, we know that the IMAT secretome is worse than visceral adipose tissue, which is crazy to think about because we all know that visceral adipose tissue is this negative adipose tissue depot. It does this in part by inflammatory cytokines, lipids, and free fatty acids, and it can induce insulin resistance in muscle. And it does this in part by eliciting an inflammatory response, although I think there's a lot more going on than just inflammation. And we know that it promotes loss of contractile proteins and size, and could be then related to decreased muscle mass and muscle force. Our data shows that the IMAT cell composition is malleable, which is good because that means that we can intervene, hopefully, with lifestyle and other measures to try to change the cell composition, which changes the secretome, which changes the negative metabolic impact. And we know that the IMAT secretome is constantly bathed in muscle, influencing metabolism. And if there's one thing I want you to kind of remember from this chat, is that the muscle is constantly being bathed by this depot all the time that is having this negative impact on muscle insensitivity, muscle mass, muscle force. And I think it's a prime target for intervention. The questions that we have now are, can we antagonize the signaling from IMAT to prevent its impact on diabetes risk, but also for sarcopenia? And the questions that we're trying to answer with the two studies that we have ongoing right now is, do lifestyle interventions change the IMAT secretome? And does that change its negative metabolic influence on muscle? So please stay tuned for that. So I am thrilled to be able to work with just an amazing team. We try to have a lot of fun together in terms of in the lab. Upper right hand corner, this was archery dodgeball, which was as silly as it sounds, and I was sore for I think 3 days after that. Of course, I want to thank my funding stream, NIH and Eli Lilly. They've both been very good to me. I have a great research team and a bunch of wonderful collaborators. So thank you all for your time. If you have any questions, love to chat.
Thank you, Dr. Bergman. That was an excellent presentation. I guess I'll just take sort of moderators first, but just, you know, I'm just wondering, I'm sure you've thought about this, but you talk a lot about IMAT signaling affecting muscle, but I can think of other, either pro-anabolic or effects of exercise where the muscle secretome could, I guess, affect IMAT. Have you thought about that a little bit, or?
Oh, yes, we have. That's an excellent question and not one I normally get. So bravo. Yeah, so what we're looking at right now is I'm at yelling at muscle. That's the way our system's set up in vitro, when it's not at all how it's going down in vivo, right? It's a conversation that's going on. So we've actually started to try to we've developed these methods where we started with visceral adipose tissue, which is a surrogate, which we have quite a lot of. And we take visceral adipose tissue and muscle and we co-culture them together for a period of time to let them crosstalk. And then we separate them and let them then have their secretome, and we measure their secretome. And with visceral adipose tissue, when muscle and visceral adipose tissue have that ability to crosstalk, it doesn't change the muscle secretome at all. Yet it makes the visceral adipose tissue much more negative when it comes to metabolic health. Now, with a collaboration we have where we get a lot more muscle with which to work, we started to try to answer the same question with IMAT. And it looks like the same thing is true. You can have IMAT and muscle co-culture together in the same well for as little as 3 hours. And then when you separate them out into their own individual cultures, that IMAT compared to a time-matched sample that has never been cross-talking with muscle, the IMAT is much more metabolically potent in terms of its ability to decrease insulin sensitivity. So now we're trying to think about, okay, what's that all about? What are these signals? How is muscle talking with IMAT that makes IMAT worse? How is muscle talking to other adipose tissue that makes those depots worse? Because I think if we can intervene there, we could actually start to intervene with some of this tissue-tissue crosstalk that's happening in vivo and hopefully have positive metabolic impacts. And
Excellent.
the data with visceral adipose tissue is crazy to me because effectively what we've shown is that visceral adipose tissue has a volume control for how metabolically negative it is. And if we could figure out that volume control, we could do amazing things to decrease negative metabolic health associated with that depot.
Great. That's really excellent. Larry Parnell asks, he says, the research that brought you and your team to fibronectin is intriguing and exciting. There is some evidence that fibronectin undergoes N-acetylglucosamine modification, perhaps relating to its function in insulin resistance. Have you observed anything in regard to this modification in the samples that you have analyzed?
Wow, that's an awesome question. And no, we have not. Yeah, so we've done O-link analysis mostly, which just captures fibronectin, and it doesn't tell us much about modifications like that. But that's a really cool thing to chase down. So great suggestion.
Great. Okay. Chris Wiley asks, he says, lovely story. A question regarding your inflammation culture data. I agree it is likely that you observed IMAT conditioned media is driving the observed effects through inflammatory cytokines, but MAPK signaling can also be activated downstream of mitogenic receptor binding through the RAS signaling pathway. Were there any non-cytokine ligand candidates in your IMAP secretome that might also drive similar responses to inflammatory receptor binding? That's a long question.
Yeah, no, it's an excellent question. And the short answer is absolutely. We have not yet done a thorough dive into our O-link data because we're trying to get these batch analysis dialed in. But there are so many potential ligands coming out of IMAT that could be causing these effects in vitro. We haven't looked at them thoroughly, but it's not just inflammatory cytokines. And in fact, one of the things that we've done is we've looked at visceral adipose tissue, and we've tried to use growth pathway analysis to kind of figure out how much of everything that is being secreted by visceral adipose tissue, and we're going to do this with IMAT as well, how much of that is inflammatory compared to other? And only about a third of what is secreted by visceral adipose tissue is inflammatory, which means two-thirds is this other, any other kind of protein that might be having these effects. So absolutely, there is a ton of possibilities by which other protein ligands outside of inflammatory cytokines could be having these effects, and we're excited to dive into them.
It's great. Andy Greenberg asks, culture of adipose tissue in culture for 24 hours alters gene expression and cytokine profile. Have you compared gene expression and cytokine profile of freshly isolated tissue versus cultured tissues?
Yeah, we looked at cytokine secretion, not gene expression. And we actually published this. This is Khan's work from, I think, 2022 or 2023. So how we got to the whole method that we have to measure the secretome is we took tissue from bariatric surgery patients and we cultured it for up to 48 hours. And every 3 to 6 hours, we then sampled some of the secretome and evaluate the secretome profile. And this was a crazy cool study, and my students volunteered for that, and they literally slept in the lab during this to get these over 48 hours with sampling every 3 to 6 hours. And what we showed was that over the first 24 hours, over the first 8 hours or so, there's very little cytokine production, and then it starts to really ramp up over the next 24. Over the course of those 24 hours, we also looked at HIF-1 alpha for a hypoxia signal. We didn't see one. It didn't change compared to fresh. We also looked at LDH as a marker of cytotoxicity to make sure we aren't just studying kind of a cytokine response. No change there either. So it looks for adipose tissue, up to actually 36 hours, it's pretty happy in culture when you follow this protocol of cutting it up into smaller bits, allowing it to actually be oxygenated, and there's no cytotoxicity. So it's actually a pretty solid approach. And so we published that so people can look at it, and other people are starting to use it now, which is great.
Okay, there's two more questions. Zhandong Wang says, great presentation, thank you. He has two questions. The increased intermuscular adipose tissue by obesity or aging is due to larger size or increased number of intermuscular adipocytes. And the other question is whether obesity or aging without insulin resistance induced IMAT differs in metabolic or inflammatory responses or mechanisms. So more cells or bigger cells? I guess that's the
Yeah.
first question.
We don't know yet. Yeah, so we're only now able to get enough adipose or intermuscular adipose tissue where we're actually going to be able to do histology to answer this question. And to give you a feeling, a lot of this data is generated on IMAT biopsies that are 4 milligrams, 5 milligrams. And so they're so small. I've never wanted to actually use it just for histology to answer that question. Instead, we're doing the secretome analysis where we get a lot more bang from our buck. But it's a great question, and we don't know, and it has a lot of implications. And so the other question was IMAT.
Um, Obesity and aging without insulin resistance induced IMAT differs in metabolic or inflammatory responses or mechanisms.
Yes, the metabolic response is different, and we've looked at that relationship with age, with the ability for IMAT to decrease insulin sensitivity. And if you do that in individuals without prediabetes or type 2 diabetes, you still see that relationship. So it seems like even if you're a healthy, normal glucose tolerant individual, as we age, it looks like our IMAT tends to be more negative. So yes.
Okay. And then last question from Dr. Greenberg. Adipocytes can secrete fatty acids that also will alter gene expression and cytokine levels. Do you control for regulation of adipocyte lipolysis that might be impacting your results or affect your results?
No, we don't try to control for it, and I don't even know how we would do that. We don't try to shut off lipolysis. We actually try to measure the rate of lipolysis as an indicator of how lipolytically active the tissues are. And so we've actually shown that IMAT is as lipolytically active as visceral adipose tissue, so high compared to subcutaneous adipose tissue, which is lower. So we don't try to control it. We just try to let whatever is happening, at least in vitro, happen so that we can kind of see what its normal instincts are, if you want to think of it that way, in terms of free fatty acid release. And it looks like it actually releases quite a lot of FFAs that can have those impacts, like you mentioned.
Great. So I don't see any additional questions. Nothing else has popped up. So thank you, Dr. Bergman, for an excellent seminar. And I think the questions were very provocative and interesting as well. So I want to thank everybody for attending this week's seminar series. Thanks so much.
Awesome. Thank you, everybody.
That was a great talk.